Hello, everyone, and welcome to today's webinar, Targeting Prostate Cancer Stem Cells with T-cell Immunotherapy. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I would now like to present today's speaker, Amy Codd. For a complete biography on Amy, please see the Biography tab at the top of your screen. Amy, you may now begin your presentation.
Thank you for that introduction. Hi everyone, I'm Amy. I'm a final year PhD student in Cardiff University. I'm in the Tissue Microenvironment Group, and my supervisors are Zsuzsanna Tabi and Stephen Mann. So I'd like to thank Thermo Fisher for sponsoring this presentation and the Cell Culture Heroes program, as I think it's a really great opportunity to present my work, and thank you also to LabRoots for hosting. So my talk today is about targeting prostate cancer stem cells by T-cell immunotherapy. So I'm going to start by giving you an overview of my talk. I'm going to be talking about each of these elements today: cancer stem cells, prostate cancer, and immunotherapy. So to introduce cancer stem cells, I want to talk first about the cancer stem cell theory of cancer development, and then also how cancer stem cells can be identified, which is not actually the easiest thing, considering the cancer stem cell theory is not a cut and dried and coherent idea. But actually, I'll go into that more in a little bit. Next, I'll be speaking about prostate cancer, as my work on cancer stem cells has been focused on prostate cancer. It's actually a big area of interest here in Wales, so I'm based in Cardiff University, and also wider in the UK. And here in Cardiff University, we have really good connections to the hospitals here for getting samples, which is really lucky. Then I'll be speaking about immunotherapy, which I'm sure at this stage doesn't actually need that much of an introduction these days, as it's very much in the news and becoming more and more commonly used in cancer treatment. So I'll be discussing the basis of cancer immunotherapy, which is cancer immunology, and how this was first proposed, so like the interactions between the immune system and a developing tumour, and then how these interactions are activated, inhibited, or otherwise modified for cancer immunotherapy. As my focus today is on T-cell immunotherapy, I'll be discussing the identification of T-cell antigens, which is something that I've done in my PhD, and then I'll be discussing how this can be applied to cancer stem cells, so identifying cancer stem cell antigens for therapeutic targeting. So the cancer stem cell theory of cancer. This suggests that tumours consist of a hierarchy of cancer stem cells and non-cancer stem cells, and it is only the cancer stem cells that are capable of sustaining tumour growth. This is because they are capable of self-renewal, so they're constantly able to maintain a pool of stem cells that remains dedifferentiated, and, for example, expresses genes associated with development and stemness. What they're also capable of is dividing to produce non-stem cancer cells. So in the production of non-stem cancer cells, this population, the non-stem cancer cells, don't have self-renewal capacity, and they're more differentiated, although there might be an intermediate population or like a gradual decrease in stem-like characteristics. But it's likely that this intermediate population wouldn't be able to sustain or initiate tumours like cancer stem cells can. So the non-stem cancer cells make up the rest of the tumour bulk, and the cancer stem cells are typically thought to be a very rare population, although this can vary between cancers, and so this feature isn't really a defining characteristic. So the presence of cancer stem cells, which give rise to non-stem cancer cells, means that there's both a hierarchy and heterogeneity in the tumour, which is also part of the cancer stem cell hypothesis. This heterogeneity in that the cancer stem cells and the non-stem cancer cells have different characteristics, and I'll be going into these more in a bit, and then there can be heterogeneity also based on the acquisition of mutations. So different cancer stem cells could acquire a mutation and pass this down to their hierarchy. There could be more than one clonal cancer stem cell mutation and hierarchy. And also the non-cancer stem cells, being transformed and genetically unstable, may accumulate mutations, adding further heterogeneity. So the cancer stem cell theory also suggests that it is stem cells that are the most susceptible to carcinogenic transformation, as they are long-lived, so in a position to accumulate mutations which would otherwise be lost before they initiated any malignancy if it was a differentiated cell. It's also suggested that gene expression signature of cancer stem cells is more permissive to cancer development. So, for example, continued growth instead of programmed death signals, although it is also suggested that the transition to cancer may have to activate stem cells from dormancy in a tissue. On the other hand, though, it's suggested that these stemness characteristics could all be gained in the process of carcinogenesis. So an actual mutation to transform to cancer would activate a gene signature. So it's possible that more committed progenitor cells and differentiated cells could become cancer stem cells, or that there's even a degree of plasticity and that differentiated cells could gain stem-like characteristics in an established tumour. And this set of characteristics, which is beneficial for the maintenance of cancer, like selective pressure, could select for another cancer stem-like clone. So it actually isn't fully agreed upon which is the predecessor cell to a cancer stem cell, although having these characteristics in common with stem cells is what informs identification and characterization of stem cells. So you might actually see in the literature different names used for cancer stem cells, and that's down to a number of factors. Firstly, the use of stem in the name might imply the origin of the cancer stem cell, and because, like I said, this isn't actually entirely agreed upon, it may not appear correct to use to call them a stem cell, a cancer stem cell. And then also the assays we use to identify cancer stem cells may not be able to capture even these agreed upon characteristics of cancer stem cells in our model. So for example, by isolating cancer stem cells from their niche in situ, this may disrupt the hierarchy and resulting in them behaving differently in our experimental conditions. And it's also been suggested that the cells which initiate a tumour, so these might be referred to as tumour-initiating cells, that these cells might just be the best survivors of an experimental manipulation rather than the actual self-same cells as the cancer stem cells that are found in the tissue. So this is actually a real challenge in this area to have an agreement on what assays for cancer stem cells actually demonstrate what we believe we're showing from an assay and how this affects identifying cancer stem cells for therapeutic purposes. So for a certain amount of clarity in this talk, I'm going to refer to cancer stem cells by a functional definition that the cancer stem cells are a distinct population of cells in tumours possessing stemness characteristics. And this recent review actually gives a really good detailed commentary on some of these features. So I do think it's important as well to contextualize these in the tumour microenvironment, the cancer stem cell features versus kind of the overall cancer features, because we could remember really that some of these features are common to non-stem cancer cells, like how the tumour microenvironment is generally immunosuppressive in most types of cancer. And obviously, while some of this contribution has to be from the non-cancer stem cells if these are in the majority. As I said, the cancer stem cells are typically relatively rare. And so the cancer stem cell, but anyway, so the non-stem cancer cells presumably actually contribute a certain amount of these sort of the survival and growth cues, immunosuppression, ability to resist. They may actually help promote and interact with the cancer stem cell niche. But the cancer stem cell theory was proposed to suggest why cancer relapse occurs. So it suggests that some cells, just any kind of cells, these must not be eradicated by cancer treatment, and this results in regrowth of a tumour. So there are just surviving cells which are failed to be eradicated by a treatment, and these cells presumably have some particular characteristics to allow them to resist treatment, as is suggested in the diagram there. And if it is the stem cell that becomes transformed to develop a tumour, then also, stem cells must be able to evade immunosurveillance, so evasion must be a characteristic as well as therapeutic resistance. So to identify cancer stem cells, we take from what is known about just healthy ordinary stem cells, in other words, the surface and functional markers of these cells. These markers have been identified for a lot of cancers, and this list, I'm sure, is not exhaustive, You can see that there are a lot of cancer stem cell markers which have been identified for hematological and solid cancers. Some markers are common to a couple of different types of cancers. So I just highlighted the functional markers in red to distinguish them. The functional markers are aldehyde dehydrogenase and the ATP binding cassette, so known as the ABC transporter family of proteins. These functional markers are known by that name because they play a specific role in stem cell characteristic. For example, the ABC transporter family have efflux of molecules, for example, chemotherapy drugs, and this is actually a functional protective role in the maintenance of the stem cell phenotype. So the stem cell function of some of the other markers, the other surface markers, may not actually be entirely known, and it's also clear that some of these proteins have different roles in different cells, so stem or non-stem cells, because they are actually widely expressed in the body. So, for example, CD44 there. So as I said, identifying cancer stem cells is based on what we know about stem cells, so it's probably not coming as too much of a surprise to know that the first cancer stem cells were identified in hematological cancers, because hematological lineage has a clear hierarchy and markers delineating different stages of it. So the hematological stem cell, which gives rise to a lineage-restricted progenitor, and that's differentiated but still has some self-renewal capacity, and this gives rise to further differentiated loss of self-renewal phenotypes. But this lineage is disrupted in leukemia, but it was with hematological stem cell markers that the first leukemic stem cells were identified. So what the authors showed here in 1997 was that the CD34 positive, CD38 negative cells, and they actually got these from the bone marrow of AML patients, so they were primary cells. These cells could initiate tumors in immunodeficient non-SCID mice. So that covers one of the characteristics of cancer stem cells, the ability to initiate tumors. They also showed that these cells had differentiation capacity. They found that the cells which they transplanted with these markers, the CD34 and CD38, when these cells engrafted and were tested 6 weeks later from the xenograft models, the bone marrow was later analyzed, and it had actually recapitulated the phenotype of the original blast cells. So you can see the percentages here of, for example, CD34 is very similar from the original blast cells to the engrafted ones, which had a different kind of the stem-like phenotype. So identification of stem cells, cancer stem cells in solid tumors has been a bit more of a challenge because there isn't always as clear a hierarchy. Although, for example, here in the colon, identification of cancer stem cells is helped by knowing the actual location of the stem cells in the tissue. So these are here in the crypt columnar cells. So rare stem cells with these markers can actually be identified quite clearly in the tissue by microscopy, and then these markers can be used to identify cancer stem cells. This has been done here on the right in showing that CD133 is a marker for colon cancer stem cells. This was single marker-based identification of the cancer stem cells, and some studies since may have used this marker in combination with others to identify a population with similar characteristics. I did have a longer list on the couple of previous slides of the colon cancer stem cell markers. But here, anyway, the CD133 cells caused tumor growth from much lower numbers than the unsorted cells, and even the CD133 negative cells didn't grow tumors at all. And then when the cells were grown under differentiating media, so addition of serum and removal of some of the growth factors, EGF and FGF, the cells which differentiated were not actually able to grow tumors. So in the next example, we have the same marker being used to identify glioma stem cells, so brain cancer stem cells. And this paper was particularly significant in demonstrating treatment-resistant characteristics and also identifying the underlying mechanism for this in cancer stem cells. So it was found firstly that irradiation of the tumors, and this was tumors from mouse xenografts, were enriched for CD133 cells, 133 positive cells, and irradiation of these and the CD133 negative cells demonstrated that the CD133 positive cells retained greater colony formation after radiation. They were still viable to grow the colonies, but then inhibition of checkpoint kinase proteins by this DBH molecule prevented the cells being able to activate DNA damage signaling and actually initiate repair after radiation. So you can see that almost no colonies grew in this condition. The CD133 positive cells had lower apoptosis and higher activation of various proteins in the DNA damage signaling response, so demonstrating that they have a greater capacity to respond to DNA damage, and this results in even more efficient repair and survival. So that is my introduction and discussion of cancer stem cells in general, what the theory of cancer stem cells is about, and how cancer stem cells are identified, and then some of their treatment resistance mechanisms. So I'm going to move on now to speak about prostate cancer, because as I said, it's actually been what I've been focusing on for my PhD. So this is part 2 of the talk. So prostate cancer is the most common type of cancer in men in the UK. It's globally the 3rd leading cause of cancer-related death in men, but it's actually really difficult to find a clear statistic about the frequency of prostate cancer mortality, because there's quite a complicated breakdown in terms of the disease course. So as you can see from the diagram here, the majority of men are diagnosed with localized prostate cancer. That means it's contained within the prostate gland. It includes up to stage 3, which is extended beyond the prostate capsule, but this without significant lymph node involvement. And actually the survival rate for this is really high, over 90%, because the treatments for localized prostate cancer are very effective, so radiation and surgery mainly. And prostate cancer development or recurrence is measured by levels of the prostate-specific antigen, which is produced by the prostate and then released into circulation. So rising PSA after successful localized treatment is indicative of biochemical recurrence. And this really isn't uncommon. It can occur in up to 40% of men treated. There's slight variations between the type of treatment they receive, if it was surgery or the radiation therapy. However, biochemical recurrence doesn't actually always lead to clinical progression. So at this stage, the mortality rate is still low because we have most of the men being diagnosed and essentially cured, successfully treated with early-stage prostate cancer. And only some of them are progressing to clinical progression. However, kind of beyond that stage of clinical progression, the prognosis is really poor and the mortality rate is high. So if metastasis and castrate resistance occurs in response to androgen treatment, so androgen treatment is the androgen deprivation therapy, is the standard therapy for advanced prostate cancer, and resistance to this occurs in a really high proportion of men, like almost all, and after expected quite a short amount of time of undergoing the treatment. So if this occurs, castration resistance and further metastases, the average survival is only 1.5 years. So metastatic prostate cancer essentially doesn't have a curative therapy. It's actually just life-prolonging treatment. And there's a lot of research going on to develop new therapies for this to treat the later stage. But I think it's really important to consider how an earlier intervention could actually alter the course of the disease. In other words, preventing perhaps most importantly clinical recurrence, which is where we consider that targeting cancer stem cells comes in. So back to identifying the cancer stem cells, what do we know about prostate development, the cell types, and the tissue lineage? Because this will inform us as to how we identify the cancer stem cells in the prostate. So the prostate consists of basal, transit-amplifying, and luminal cells. The basal cells is where the stem cell population resides, but prostate cancer itself actually has a predominantly luminal phenotype. And that's what you would see on microscopy staining of biopsies. It's pretty much all luminal cells. But anyway, this guides us then towards the basal markers for the identification of prostate cancer cells. The first prostate cancer cells were identified by this basal phenotype profile, CD44 positive, alpha 2 beta 1 high integrins, that is, and CD133 positive cells. So the cells with this profile were shown to have greater colony formation, colony forming efficiency than transit amplifying cells, which are the ones in the middle of that graph there, which don't express CD133 but do express CD44 and the alpha beta integrins. and then the cells which are negative for these basal integrins on the right of the graph there, these are the remaining population, presumably the luminal cells, and you can see that they don't really have any great colony-forming efficiency at all. So in a follow-up publication from the same group, they also showed that the prostate cancer stem cells sustained much less DNA damage, which is shown here by gamma histone H2AX staining, and then compared to the transamplifying, the TA on the graph, and the CB, so the committed cells, the committed differentiated cells. And a histone deacetylase inhibitor, HDAC inhibitor, reversed the radio resistance effect. So this is actually another demonstration then of the mechanism of treatment resistance by these cancer stem cells in prostate cancer. So that previous group, sorry, I'll just go back to this slide a sec. This group uses the model, they use primary prostate cancer cells derived from biopsies from patients undergoing primary treatment for prostate cancer, surgery, so prostatectomy, removal of the prostate, and then you can get cells from the biopsies. But primary prostate cancer cells are actually really difficult to grow in vitro. This is from personal experience. It's quite confusing, actually. Nobody really has a very good answer as to why they're difficult to grow. So there is a heavy reliance on prostate cancer cell lines, even though there are actually only a few of these available, and these are all being isolated from metastases. So in contrast to the primary cells, which are generally from the localized cancer, the prostate cancer cell lines are from metastases. So that is just something to consider, that different markers could be applicable in different models. So in this paper that I'm discussing now, they used the functional marker aldehyde dehydrogenase and instead the DU145 prostate cancer cell line. But they did similarly show that the aldehyde dehydrogenase high cells could initiate tumors at a lower frequency than the aldehyde dehydrogenase low cells. And they showed that they did have some primary samples as well. They showed that the aldehyde dehydrogenase isoforms, it's quite a large family with lots of different isoforms. I think there's 19. These isoforms, some of these were upregulated in response to radiation. So this could represent enrichment in response to radiation or possibly also a protective response. Although actually the signaling response that this group identified in response to radiation was that there was greater AKT2 and checkpoint 2 CHK2 signaling in the aldehyde dehydrogenase high cells. So perhaps similarly to the glioma stem cells that I mentioned before, these cancer stem cells could have a more efficient DNA damage signaling response. But here it was actually the Wnt TGF-beta signaling that was crucial to the radiation response. And there was no difference in tumor-free survival in mice in the aldehyde dehydrogenase high mouse xenografts. Whether or not the initiating cells were irradiated or non-irradiated didn't make a difference to the mouse survival, whereas there was a significant difference, an increase in survival in mice who were inoculated with irradiated aldehyde dehydrogenase low cells. So they were not really able to, they were much more affected by the radiation. So these two studies have dealt with, the first study dealt with basal-based identification of prostate cancer stem cells. And this study here using aldehyde is kind of a phenotype-free, perhaps sidestepping the issue of pinning down the phenotype of the prostate cancer stem cells. But we have been focusing on the basal cells as being the source of stem cells in the prostate. However, a number of more recent studies have identified luminal prostate cancer stem cells. So firstly, luminal stem cells just from a healthy prostate were identified. So these cells were seen to have multipotent features. So when prostate cells were isolated from a biopsy and fractionated into basal or luminal populations using CD49F for the basal population and CD26 for the luminal population, both the basal and luminal cells were capable of growing organoids in this defined media, and these organoids actually expressed both basal and luminal cytokeratins and also the androgen receptor. So each of the basal and luminal cells was able to create, to give rise to a fully functional lineage of cells. So this finding really puts a whole new spin on what's known about the cell lineage of the prostate. it does suggest a certain amount of plasticity or even multiple lineages having stemness capacity, although the authors did note in this paper that the luminal cells had a much lower organoid growth efficiency. So it may still be that the basal cells are the predominant cells of the prostate, but luminal cells may also have this capacity then under certain conditions. So another group showed in a mouse model of prostate cancer that a small subset of cells expressing NKX3-1, these cells didn't express basal markers, but these were resistant to castration. So what we actually know about the hierarchy of the prostate is that we know it was originally demonstrated by castration of rats, in which it was found that castration caused regression of the luminal cells, but preservation of the basal cells. So castration was removing the androgen signaling, and then when the androgen signaling was restored, and this caused regrowth of the prostate, the luminal cells were the ones regenerated. So this gave you the idea that basal stem cells give rise to luminal cells with the growth stimulation from androgen. So the significance of this study is that there are non-basal cells surviving castration, and this study then also showed that in a PTEN knockout, so it's a tumour suppressor on a genetic background for the development of prostate cancer, in which the animal was castrated, so removing the luminal cells but leaving the NKX3-1 cells, that these cells were drivers for cancer growth as well. So this paper actually suggested that luminal cells could be the target for transformation for prostate cancer as well. So these all show evidence then that there can be basal prostate cancer cells with the phenotype CD44 alpha 2 beta 1 integrin high and CD133 positive. Aldehyde high prostate cancer cells exist. Then there are luminal prostate cancer stem cells seen in this mouse model. So actually in my PhD I used a combination. I used the D145 cell line and primary samples from biopsies taken from prostatectomy samples. I grew these in different media because, as I said, the primary cells are quite difficult to grow, so the media needed a lot of supplements for success of it. So what I'm kind of showing here is that these cells, using a cell line and primary cells, which by appearance, by the morphology here, you can see they grew quite differently. They were under different media conditions, required this to actually grow it. The D145s have this, what's known as a characteristic cobblestone morphology, while the primary prostate cancer cells grew in a morphology which I would say is more similar to the original tissue. They didn't really grow in a flat, uniform sheet. So these are two different models in which the cells grew with a characteristic appearance of either the cell line or perhaps the original tissue. And I actually found a different phenotype in these cells. So what I'm suggesting, kind of by showing these different studies which have different phenotypes to identify prostate cancer stem cells under different conditions, 2D, 3D animal models, is that the conditions selected for the investigation of prostate cancer stem cells can bias you towards having a different phenotype in your experiments, and that this can result in differences in identification of prostate cancer stem cells. Because what I saw here was that my D145s had a different phenotype to the primary prostate cancer cells. The D145s were, as you can see, over 2/3 basal cells, so CD49F positive, but there was a double positive population for CD49F and CD26. And I think this suggests an intermediate population, which may correspond to transit amplifying cells. But on the other hand, my primary prostate cells grew with an almost completely basal phenotype. So for this reason, I didn't use a lineage-specific approach to isolating the cancer stem cells. I used the functional marker aldehyde dehydrogenase. And I was almost surprised to find actually that the aldehyde dehydrogenase high frequency of population in the D145 and primary prostate cancer cells was quite similar proportions. So this could suggest that the cancer stem cells in this model, this specific model, do reside in the basal compartment of the cells, seen as I had a similar aldehyde frequency in the absence of the intermediate phenotype in the primary cells. But this, I think, really emphasizes how identifying particularly prostate cancer cells and the markers used for this is quite dependent on the conditions and the model used. So moving on to part 3 of my talk, where I'm going to be discussing how cancer stem cells can be therapeutically targeted. So we have established that if cancer stem cells are not specifically treated, or if a conventional treatment that the cancer stem cells are resistant to is unsuccessful in killing the cells, this can cause treatment recurrence. So a treatment for cancer stem cells then must be able to kill them in a different way that they might not be resistant to. And it must be specifically targeted so that we know we're taking out the cancer stem cells. So this could be inhibition of pathways intrinsic to cancer stem cells, like developmental signaling, or by immunotherapy targeting, which is what I'm going to focus on for the rest of the talk. So before I get discussing actually using the immune system to treat cancer, I do want to speak about why this actually works, in that the immune system is capable of recognizing and killing cancer. So this ongoing process is described as the 3 E's of immunoediting. So the immune system is capable of controlling cancer development, and that's known as elimination. However, if it isn't fully eliminated, a premalignant growth may interact with the immune system over time, and this is known as equilibrium. This process can end in elimination, which is great, or suggest for genetic immune escape variants and aggressive growth. So then escape describes cancer growth, which is a failure of the immune system to contain it. So immune cells with an anti-tumor role include CD8 and CD4 T cells, NK cells, and inflammatory M1 macrophages. However, in a tumor which has escaped the immune system, immune cells could actually have a tumor-promoting effect, which includes T regulatory cells and M2 inflammatory macrophages. These cells inhibit actually the tumor-clearing immune response, and the tumor itself doesn't also have a passive role in this process, so it can express inhibitory receptors, produce anti-inflammatory cytokines, and this makes it more difficult for the T cells to respond by downregulating. The tumors can also make it more difficult for the T cells to respond by downregulating antigen expression, so downregulating HLA expression levels. So immunotherapy then aims to introduce or boost a tumor-killing response by engaging the anti-tumor immune cells that I mentioned, or actually by blocking some of the inhibitory signals. So cancer immunotherapy was Science Magazine's Breakthrough of the Year in 2013, and I'm sure most of you will have heard how the discoveries relating to the mechanism of action of checkpoint inhibitors was awarded the Nobel Prize last year. These are the main types of immunotherapy. So checkpoint inhibitors refer to blocking some of the immune inhibitory signals I mentioned, and these, I would say in particular, are at the forefront of the promising results around immunotherapy, but also there are many other areas. So monoclonal antibodies are being used more and more as receptor agonists or antagonists in crucial cancer signaling pathways. Vaccines are being developed, for example, which engage dendritic cells to provide the required signals of an immune response and direct it towards a cancer antigen. But on the subject of cancer antigens, I'm going to focus today on adoptive T-cell immunotherapy, as that is what I have been investigating during my PhD. So CD8 T cells are the killer cells of the adaptive immune system. So they kill cells in an antigen-specific way by releasing effector molecules, perforin and granzyme, into the target cell and activating death receptor signaling. T cells are capable of rapidly proliferating when they're activated, so a large number of T cells can respond to an antigen stimulus. Although in immunotherapy, this activation and co-stimulation might actually be done ex vivo, so by maybe extracting the cells from the patient or modifying cells from healthy donors, because the correct stimulation activation response is often disrupted in cancer. And the natural course of a T-cell response in the body includes the persistence of antigen-specific memory T cells. And I would say while this hasn't been sort of demonstrated, the persistence of memory having been shown in an experimental condition, it is actually suggested to be one of the main reasons for the durability of responses observed with this form of immunotherapy. So really importantly, T cells can respond to an enormous range of intracellular and cell surface proteins by recognizing the short fragments of the protein known as antigens, which are presented in complex with HLA at the surface of the cell. And identifying tumor-associated antigens is one of the main focuses of T-cell immunotherapy. And this can be done by predictive or experimental methods. So examples of these predictive methods, DNA library screening involves large-scale cloning and production of proteins from patient samples, and immunological, so either antibody or T-cell screening of proteins for an activation response. So a number of prominent clinical targets have been identified in this way, for example, NY-ESO, MART1, MAGE1, and then an alternative way is using reverse immunology, which involves in silico prediction of antigens using algorithms. And these algorithms can actually predict pretty much the course of a protein's journey to antigen presentation, including proteasomal processing, tap transport, and HLA binding, although it does have the downside that it might not actually identify sort of the most immunogenic antigens. There's not really any way of knowing how immunogenic your antigen is until you go and test it in an experimental condition. But at this web link then is one of the ones that I've used quite commonly. This web page has different algorithms for testing these different aspects of predicting antigen binding. But antigens can also be identified experimentally by capturing them in complex with HLA from the cell surface and then uncovering the epitope sequence by mass spectrometry. This is known then as immunopeptidomics or HLA ligandome analysis, and this is what I've mostly done during my PhD. So the way this is done is that antigens are captured by immunoprecipitation of the HLA complexes from the surface of the cells, and this can be sort of tailored to a specific HLA allele if you have the antibody against that allele, or actually broadened to include capture of any HLA class 1 antigen on the surface of the cell. And this area is really growing because it enables novel antigen discovery. This really interesting paper that I'm referring to here is about machine learning to improve antigen prediction from real mass spectrometry data sets. And then this can all help increase our understanding of antigen processing by analyzing the data sets, which can feed back into improving predictive methods. So the HLA ligandome analysis in this example here was used to identify a cancer stem cell antigen in colon cancer. So this group actually established a cancer stem cell line by repeated cloning of side population cells from a colon cancer cell line. So side population cells, that's one of the sort of functional assays for cancer stem cells, because these cells have highly active ABC transporters, which efflux the host dye. So it's a functional method for isolating the cancer stem cells. So HLA ligandome analysis of side populations, so the cancer stem cells, or main populations, so non-effluxing or non-stem cancer cells, did reveal differences in antigen presentation, including this one that they've identified here, the IV9 peptide of the ASB4 gene. So this peptide was actually found exclusively in the HLA ligandome of the side population or colon cancer stem cells compared to the main population. So a T-cell clone was developed which responded to the IV9 peptide pulse cells with little off-target specificity up in the kind of top line here, the graph A on the top row. And these cells actually preferentially killed side population cells in B, the specific cancer stem cell targeting. Then in vivo model investigating tumor control and adoptive T-cell immunotherapy, essentially, these IV9 specific T-cells were capable of presenting tumor growth, actually whether given prior or after tumor challenge. So I was really lucky to have the opportunity to collaborate with the group that produced this paper. They're from Sapporo Medical University in Japan, and with them I carried out HLA ligandome analysis to identify novel prostate cancer stem cell antigens. We did have a bit of a different approach. We didn't establish a cancer stem cell clonal line, as this is quite time-consuming and definitely a challenge to maintain a pure and stable clonal stem line. So what we did instead was we analyzed the DU145 prostate cancer cell line, the whole population, and then we identified cancer stem cell specific antigens based on the gene expression of the antigens in sorted aldehyde dehydrogenase high or low cells. So I was saying that the aldehyde dehydrogenase marker was the one I used during my PhD project to identify the prostate cancer stem cells. So we performed PCR of some of the genes of interest corresponding to the antigens and looked at the ones which were more highly expressed in the aldehyde dehydrogenase high cells. So we identified these targets, as I said, in the D145 cells, but actually we did also see, because I was using primary prostate cancer cells as well, we looked at which antigens had a higher gene expression in the aldehyde dehydrogenase high primary prostate cancer samples. So these are actually the graphs that are shown here. So the graph on the left demonstrates one of my cancer stem cell specific antigens of interest, and the graph on the right is what we designated shared antigens, for which the gene expression was not different exactly between the aldehyde dehydrogenase high and low cells, but it was there was a high abundance of the gene transcripts in the PCR analysis, because we felt that these would be a relevant target, being high gene expression, hopefully high protein turnover and presentation at the cell surface. So still being a relevant target for both cancer stem cells and non-stem cancer cells. And in any case, we also felt that from a sort of translational point of view, that an antigen-based immunotherapy is probably more likely to succeed by targeting multiple antigens and preventing the possibility of antigen escape variants. So my final work in my PhD has been on the isolation of T-cells which recognize these antigens, and perhaps unsurprisingly, the frequency of T-cells responsive to the cancer stem cell antigen was really very low, but I have actually been able to expand these cells quite a bit with the goal of developing a clonal T-cell line. I was also very lucky to have a donor that was the correct HLA type, which is B50, which is in the U145 cell line. It is a lesser common HLA type, but I was very lucky to have a healthy donor who was the correct HLA type. And the shared antigen that I mentioned on the previous slide on the right here, so gene number 3 or antigen number 3, this was identified from the D145 cells, so it was a B50, HLA B50 epitope. However, it isn't actually what I eventually isolated T-cells against. You can see I have a HLA-A2 donor here. So actually, when we modeled the B50 epitope interactions in a homology modeling design, we found that it didn't really have particularly optimal interactions in the HLA binding pockets. So instead, we identified a HLA-A2 epitope from the same protein, and obviously had a much higher abundance of HLA-A2 donors to test, and we were actually able to decide which donors to use to develop the T-cell lines from, because we had, as you can see here, quite a good response from this particular donor. So there are lots of other studies investigating immunotargeting of cancer stem cells, which have progressed to in vivo models and look very promising. An example here is in prostate cancer, where this group developed CAR T-cells targeting the cell membrane protein EpCAM. This is a CAR T-cell, so it's a different approach to adoptive T-cell therapy. It's against surface antigen or surface proteins. So non-HLA restricted or presented antigens. This is actually a protein which is expressed on the cell surface. So the CAR T-cells were tested against xenograft tumors initiated by 2 different prostate cancer cell lines. So the first one had widespread and high EpCAM expression, and the other one actually had a rarer population of EpCAM expressing cells. And the CAR T-cells were effective at controlling tumor growth in the high EpCAM expressing model, which was great and very exciting. But even more interestingly, the CAR T-cells were able to prevent metastases in vivo just by targeting the smaller proportion of EpCAM expressing cells in the PC3 model, the PC3 prostate cancer cell line, which was used in the xenograft model shown here. So the alternative one was a different PC3 related cell line with the higher EpCAM expression. But shown here is the ones with much lower EpCAM expression. As you can see here, there's very little metastases over time. So there is good tumor control from these CAR T-cells. Another study then here targeting cancer stem cell antigen was actually done by adoptive CD8 T-cell immunotherapy, and this was targeting an epitope of aldehyde dehydrogenase. This study looked at a couple of cancer type models, including shown here head and neck cancer breast cancer, and pancreatic cancer. The aldehyde epitope was identified in a previous study by mass spectrometry analysis of peptides eluted from tumor lysates from a head and neck cancer patient. And as you can see here, the T cells developed to recognize this antigen were capable of reducing tumor growth in the head and neck cancer model, and also greatly improved survival in a lung cancer metastase model of breast cancer. And these show that, as well as identifying novel antigens for cancer stem cell therapy, like in the previous studies, my own work and the previous studies that I showed, targeting antigens which are identified from just cancer stem cell protein markers, this can also be an effective therapeutic approach. Although I will say that management of off-target effects in using the same markers of healthy cancer stem cells as the immunotherapy is actually a really important factor to control against. So if the markers used are common to healthy cells, you do want to make sure that the T cells don't go off and target them and cause undesirable side effects. So I'm summarizing now. I hope I've shown you today that immunotherapy of cancer stem cells is a viable and effective approach in preclinical models. I think cancer stem cell immunotherapy is actually also reaching early stage clinical trials, so it's a really promising and exciting area of cancer research. So summarizing here, cancer stem cells are the main drivers of tumor growth and are present in most forms of cancer, and they are important therapeutic targets. This is particularly relevant in prostate cancer to prevent recurrence and progression to incurable disease. Immunotherapy, which is being widely tested and implemented for cancer treatment, represents a way in which cancer stem cells can be treated in a specific manner that they might not be resistant to. And treating cancer stem cells by T-cell immunotherapy requires identification of antigens, and a really effective way of doing this is by immunopeptidomics. And I did this in my project. I used the HLA ligandome analysis to identify prostate cancer stem cell antigens, and we can see then other studies have also demonstrated effective outcomes in preclinical models of cancer stem cell immunotherapy. So these are my references. I've referenced a lot of really interesting studies, and I'm sure there are many other publications in this area that I haven't been able to include for reasons of time constraint. And last and most importantly, really, are my acknowledgements. So I've been really lucky to have had excellent supervision during my PhD. In particular, I am really grateful for all the time invested by my primary supervisor, Zsuzsanna, and also Steve's input. I've had the opportunity to collaborate with so many people with such an amazingly wide range of expertise, which are right on my doorstep in Cardiff University. As I said, I performed the ligandome analysis in collaboration with Sapporo Medical University. I was hosted there very generously in 2017. My PhD is funded by the Welsh cancer charity Tenovus, and for the trip to Japan, I also received support from the Great Britain Sasakawa Foundation. And my primary prostate cancer work, the samples were made possible by the Wales Cancer Bank. So thank you again to Therma Fisher and LabRoots and Alex, one of the previous speakers, for nominating me. Some of my social media details are here too, so I'm happy to discuss things further via these channels, and I'm happy also to continue taking questions now. Thank you.
Thank you, Amy, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions that we have time for. Okay, let's get started. Amy, our first question is targeting antigens that are specific to cancer stem cells or shared in both cancer stem cells and non-stem cancer cells likely to be better therapeutic approach?
So that's a good question. And as I said, we did look at the two possibilities, or we identified two different either cancer stem cell specific or shared antigens. So there's the group that I worked with, actually, they have a publication discussing this, and the group in Japan, and they do kind of speculate that if you have T cells which are specific for a cancer stem cell antigen, that T-cell clone in a therapeutic situation, there would be a better effector to target ratio to eliminate the cancer stem cells than comparably having the same one T-cell clone, which would have to deal with both non-stem cancer cells and cancer stem cells if it was a shared antigen. And they do think that it might be better to have the higher effector to target ratio. However, an overall more effective therapeutic approach would probably be to have different T-cell clones dealing with both elements, or combine a therapy which otherwise eradicates the non-stem cancer cells, like for example radiation therapy or some of the more other personalized therapies. So yeah, I hope that answers the question.
Yes, thank you, Amy. Now, next question: how early is it possible to diagnose prostate cancer with proposed method?
So what I've really discussed here is more of a treatment method than a diagnosis method. The standard ways of diagnosing prostate cancer, they include PSA monitoring, so the prostate-specific antigen that I did sort of briefly refer to as a monitoring for recurrence. That's also used it's not rolled out typically in screening globally, but it's used as a monitoring method to be roughly predictive of prostate cancer, and prostate cancer then is ultimately diagnosed by a biopsy. As to a treatment method, ideally we would believe that it would be most effective to be able to treat cancer stem cells in a primary therapeutic approach in order to prevent relapse. As I said, it's quite a highly prevalent problem in prostate cancer. However, it would of course still be relevant and needed really to find an immunotherapy which treats metastatic prostate cancer. Currently there's only one licensed therapy for metastatic prostate cancer called T. So if the cancer stem cell clone is also metastasis initiating, it would certainly be relevant to test anyway in the first instance cancer stem cell specific T-cell immunotherapy for metastatic prostate cancer. But it would be still very important, and I think, to consider sort of the clinical approach of not over-treating, which as it is sort of a bit of a problem in prostate cancer, to try and predict the patients who are going to progress and have the metastatic aggressive disease, which is the main reason we would want to be we would be trying to prevent that or to treat it.
Thank you, Amy. Our next question: do you grow the primary cells hypoxic conditions?
No, we don't. That's a really good question, and it's very relevant because there's a lot of kind of research into the cancer stem cell niche, and I think I probably alluded to it a bit in my talk by saying that sometimes different markers might be more relevant for different cell lines, different media conditions. Hypoxia is a very relevant one condition for cancer stem cells because they are thought to sort of thrive actually in a hypoxic niche, and unfortunately I simply didn't have access to the infrastructure to grow my cells in a hypoxic niche, but it would be definitely very relevant to look into that in future investigations.
Okay, Amy, we have time for just a few more questions. Our next question: is the discovery and targeting of cancer stem cell antigens applicable to other types of cancers?
Yeah, for sure it is. So I did refer to the one in colon cancer in the presentation. It has been done in a couple of other types of cancer from the point of view of novel antigens, as opposed to just knowing what the known cancer stem cell markers are and deriving antigens from them, like the other paper that I mentioned for aldehyde dehydrogenase. So it's actually been done in a couple more by ligandome analysis in AML, CML, and also quite recently in glioblastoma. And I can probably provide some references there if there's further interest in that.
We have time for one more question, and I want to remind our audience that any questions we did not have time to get to today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. Now, Amy, do you feel the primary cells capture the heterogeneity of the cancer you described better than the cell lines?
To a certain extent, yes, I think so. So from the morphology, I don't know how clear it was really, the image in my slide, but certainly I would think in the morphology point here, the primary cells definitely in appearance grew in what looked like more reminiscent of sort of the slices of biopsy that you would see, as opposed to just being a more homogeneous flat adherence to the plastic of the D145 cells. They also had a much shorter lifespan, so they weren't immortalized. They grew for kind of 8 or 9 passages at most, and I did also find that there would be fibroblasts growing out of them, so obviously they do actually retain some plasticity there. But then again, you know, from the phenotype that I showed, neither of these cells did manage to grow to have any luminal cells present, which would be the predominant form of cells in prostate cancer. So, you know, I would still acknowledge that it's not exactly a perfect model there either. But at the same time, we found with the basal sort of guidance, the aldehyde dehydrogenase phenotype, sort of a little bit like I'd said from one of the other papers, sidestepping slightly the phenotype issue, that we were able to still find aldehyde dehydrogenase high cells, which I did confirm in a couple of other assays that they had some cancer stem cell characteristics.
Thank you, Amy. Do you have any final comments for our audience?
No, I don't think so. Thank you, everyone, for attending and listening, and please do feel free to reach out if you have any further interest in the area. Some of my social media handles there. And no, that's it. Thank you. Thanks so much.
A big thank you again to Amy Codd for her time today and her important research. We would also like to thank LabRoot and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through July 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.